Welcome, everyone. I appreciate everyone that's joined us today, both in person and online. Today, we have an outside guest to speak, but from the NIH, from our North Carolina branch at National Institute for Environmental Health Sciences. Alison Motsinger-Reif will be joining us and presenting her work from NIHS. And she's done a lot of work at the intersection of genomics, environment, and a number of different variables. She is co-PI of a cohort called PEGS, which I should have actually looked what this stood for, but she's probably going to mention it. And it's an environmental collection of about 20,000 people in the North Carolina area that also has genome sequence data. It has a deep environmental exposure data. And she's looked at that data in a number of different ways, which she'll talk about. She's also leading a study that is an ancillary study to all of us, which we'll talk about, which is gathering deep exposome data to link with all the other types of data we have to look at the risk of incident type 2 diabetes in the environment. She is their chief of your branch, which is the Biostatistics and Computational Biology branch at NIEHS, and been a great collaborator in these projects. So, Alison, I'll turn it over to you.
Thank you so much. Thank you, Josh, for the invitation. Thank you, everybody, for their time, especially as many things are going on right now. I know everybody's time and attention is even scarcer than normal, so I'm really grateful to get this time with you guys. I'm going to attempt to share my screen now, and hopefully we'll make that transition just fine. All right, do you guys see one big slide? Excellent. Love it when the magic works. So as Josh said, I'm a human geneticist at NIEHS. I come from a human genetics background. Josh and I are both Vandy alums at different times. So I think I'm supposed to say anchor down to him. But that slang is after when I actually left. And so it's been really fun. Like I said, coming from a human genetics perspective, I was a faculty member at NC State for a long time in their statistics department and bioinformatics research center. And then I joined NIEHS about 6 years ago, right now for the leadership role in the branch, and have had the wonderful opportunity to work with this study that Josh mentioned. PEGS stands for the Personalized Environment and Genes Study, and I'll tell you a little bit more about it as we go along. So overall, I've gotten away with in my career sort of a mix of methods developments from a statistical, biostatistical, computational biology point of view, as well as really doing a lot of applied work. And the balance of my portfolio has changed through the years, but really enjoying doing both of those things. I'm not that clever a statistician. I'm not going to be in my office deriving the asymptotics to anything new. I steal all my methods challenges from the real data that I'm working on. So I'm always really transparent. And I guess if you highlight some themes, I've worked in machine learning, pharmacogenomics, thinking about gene-gene and gene-environment interactions, dose response modeling, and really I'll deal with whatever ohm is huge and popular and challenging. So this data I'm going to talk about now has been a real fun way to really think about diverse data. I hope it's okay. I'm going to take you a little bit on a whirlwind tour here and appreciate your patience. I'm going to tell you some of the things we've been doing over the last 2 to 3 years with this study that I've mentioned from a number of different angles. Sort of the studies I'm going to talk about are prioritized based on mostly fellows and trainees' interest and where they want to go in their career, as well as what we think is going to be scientifically impactful. And as I mentioned, I'm going to talk first about the work we've been doing in PEGS, and then we'll end the discussion with some of the work that's ongoing in the partnered research study that Josh mentioned with all of us. And I hope you'll be able to see the connection between some of the kinds of work and the way we've been thinking about things in the PEGS data and how that can be really applicable in the types of data that all of us is now really thinking about collecting that I think is really exciting. So I hope you're willing to hang with me. We'll move around across topics. So I'm going to use the phrase we very generously, right? Like any good PI, I actually don't do anything. Everything that I present as we is from my really talented group of folks that have done the bulk of this work. Dr. Frida Akhtori is a contractor staff scientist. Dylan Lloyd is a grad student that just graduated and will be moving to Harvard next month for a postdoc. John House is a staff scientist. Jasmine Max, another graduate student that'll defend this summer. And then Ryan Campbell is another contract staff member, and Joe is a postdoc. So really, I'm going to take credit for all their work, as well as I have to go in and highlight other key co-conspirators that helped me get away with all of this fun stuff. The folks collaborating in PEGS, Jan Hall has been the co-PI of PEGS. She's retiring next month. Dave Fargo, Charles Smith, and Kyle Messier both are all fantastic bioinformaticists, computational scientists, tenure track investigators. Then I'll highlight Jeff and Josh and others at all of us that have really been the co-conspirators on the ancillary study. Dr. Rick Wojcik, our institute director and one-on-one collaborator at NIEHS that has supported all this work as well. So I'm just going to sort of introduce here. I don't know what my audience is on the mix of geneticists versus other interdisciplinary scientists, but I want to just sort of cue up what we're doing within sort of an exposomics framework. So like I said, I was trained initially as a geneticist, so right, I'm certainly used to thinking about what's the genetic etiology of common complex traits. But as I did more pharmacogenomics and moving more and more into environmental health sciences, really am appreciating how much more information there is to gather from an exposomics standpoint. And so when I say exposomics, I'm going to be really, really inclusive in that. So here I mean things like that you probably think about as environmental exposures. What's your physical environment? How much air pollution is near your house or near your work? What sort of chemicals do you use in your job occupationally every day? So really broadly. But I also want to make sure we include in that the social environment, whether there's social determinants of health or other sorts of social factors that control the body's physiological response. Things that you might consider lifestyle, diet, exercise, sleep, that kind of thing. And then also within exposomics, the body's response. So how does the body manage that? Whether that's exposomics, metabolomics, right? How does the body process these chemicals that they're exposed to, et cetera. And hopefully you'll see I'm very inclusive in that and sort of an exposomics approach. Exposomics data to me, and the way I'm going to use it, can come from multiple sources. So that could be things like survey questions that I'm going to talk a lot about, or that could come from geospatial estimates of exposure, right? We've got estimates of air pollution or other sorts of exposures. That could include, sort of omics data, right? Metabolomics, exposomics as a proxy for your environmental exposure. So try to think about that really inclusively. So now, hopefully you're sort of queuing up to the framework, and I'll give you some more details on the PEG study and talk about what we're doing with it. So as Josh mentioned, it's a relatively small cohort, especially from a genomics perspective, but it's a decently large cohort from an environmental health sciences perspective. And we've spent the last 5 or 6 years really gathering these multiple data types in the study. So we've got a lot of data on genetics. Over the years, this cohort has been around for almost 20 years, and there's been some haphazard sort of candidate gene collection along the way. But a major effort a few years ago to collect whole genome sequencing data on everybody we could afford at the time, which is about 4,700 individuals. We recently, last year, were able to get methylation data, epigenetic data from the EPIC chip on everybody that we have whole genome sequencing. For phenotypes, we have a number of self-reported diseases and conditions from our questionnaires. And I'm going to tag in more detail on our questionnaires when I talk about the environmental exposure. So PEGS has huge questionnaires, patently absurdly huge questionnaires compared to what's often collected. So we have 3 questionnaires, one we call health and exposure. And this gets to a lot of, has a doctor ever diagnosed you with A, B, or C? And then in your work or in your home, have you ever been exposed to X, Y, or Z? A lot of questions stolen from well-established instruments, studies like NHANES, Nurses Health, etc. And then we have 2 other surveys that were taken by fewer individuals, but really extensive on what we call our internal and external exposomes surveys. So the internal exposome, you could probably relabel lifestyle, and that fit pretty well. So what medications are you taking? What physical activity do you participate in? That's sleep, stress, that sort of thing. Then the external exposome has a lot of detailed residential and occupational exposures. And then we also have a continuously growing and really dynamically growing right now set of exposures from geospatial linkages. So we have addresses and address histories for all of our participants in PEGs, including things like the longest lived childhood and the longest lived adult address, where we've linked a growing number of exposure estimates. Just to give you a snapshot of some of the demographics of PEGs, the average age when they took the health and exposure survey is about 50. We've really got some pretty broad diversity in terms of education levels. You can see some graphs there. We are mostly a Caucasian ancestry cohort, about 60%, but almost 28% come from African American ancestry, which is decently unique, actually. We've got more women than we do men. It's a volunteer cohort that happens pretty steadily. And then we do actually have a pretty good diverse range of sort of incomes and other sort of measures of socioeconomic status. Just to give you a couple snapshots on those different surveys. So in that health and exposure survey, we asked questions about over 120 sort of clinical conditions and disease states. We've got some snapshots here. You can imagine we have sort of about population prevalence of most of the common complex diseases. We're slightly healthier than the North Carolina population, but not by much. And like I said, to give you just a snapshot of some of those lifestyle factors as well. Then I mentioned these exposome surveys, our external and internal exposome, over 200 questions each. Exposome has things like the characteristics of your current and past homes, workplace characteristics, chemical and metal exposures, hobby exposures, and UV light exposures. And then internal exposome has medications, vitamins, supplements, drug treatments, physical activity, stress, infections, sleep, diet, sort of sibling twin birth order, genetic history. In total, between those 3 surveys, our participants actually answer about 1,700 questions, which is anybody's ever been involved in sort of that design, that's a lot. So it's really, really rich survey questions. Then I mentioned our GIS data. We've got a growing number of data layers there. Our first linkages were to what I've learned is actually a technical term, environmental bads. So it's how far away do you live from these environmental bads, things like airports. CAFOs are caged animal feeding operations that are not entirely unique, but a North Carolina specific exposure. And then things like cell towers, drinking water, dry cleaners, hazardous waste sites. You can see the list here of just a distance to these environmental bads. For again, all of our addresses now and longest lived adult and then longest lived childhood, if that data is available. The GIS data, we've got a growing number of linkages. So we've done a lot of linkages to measures of neighborhood socioeconomics and structure, a lot to air pollution, whether that's wildfires or PM 2.5. Again, a growing number of those point source metrics, distance to environmental bads. We're working on more and more particulate matter linkages, a lot of high resolution land use and land covers measurements, things like green space, urban density, a number of climate and climate change measures, including temperature extremes and seasonal variability, and a lot of sort of linking with public open data API sorts of things. We have great coverage over the entire state of North Carolina. We definitely are biased to sort of the Research Triangle Park area here where NIEHS is located, but we have participants in 99 out of the 100 counties in the state of North Carolina. I also am from North Carolina. This is where I grew up, so it has lots of sort of personal resonance for me. Now I'm going to start this whirlwind tour of some of the things that we have worked on and thought about. So I hope this, helps show the kinds of things we're thinking about. So former postdoc, Dr. Eunice Lee, led some work. She's really passionate about cardiopulmonary outcomes and wants to do that for her career, and led what we call an XWAS on sort of the atherogenic cardiovascular disease outcomes. So XWAS, I'm meaning exposome-wide association study. You can think of this as analogous to a GWAS if you're familiar with that area, but we're really in a sort of a hypothesis generation and sort of big data exploration space. So she took all of our exposures from the questionnaires, that over 1,700, and did association analysis where she tested, exposure by exposure with association with one of several atherogenic cardiovascular outcomes. I'm showing you here a tile plot. So on the bottom x-axis, you see the exposures, that question. On the y-axis, you see the cardiovascular outcome that it's associated with. And everything that has a colored thing on this tile plot had a significant association after a pretty rigid FDR multiple testing control. The top actually has the effect sizes of those, and the bottom just has it magnified by the strength of association. So doing this, found some interesting results. We were able to reproduce a lot of things that make us feel sort of solid about the cohort and the results we're finding, and found some new things that hadn't been reported before. In particular, strong associations with blood type A, Rh negative, so A negative with heart attack, paint-related exposures with stroke, biohazardous materials at work with arrhythmia, and strong associations with maternal education level and several of those outcomes. So you can start to see, we can start to explore what are shared environmental associations and what are unique, and sort of look across that space. You'll see that we've scaled up this XWAS approach to multiple outcomes following her work. We've also done some very targeted sort of gene environment analysis work. So here we worked with candidate genes. So we were interested in what's the association of the sort of distance to these CAFOs, we call them, these caged animal feeding operations, which are really, they're what they sound like. They're very dense industrial agricultural sites that have absolutely terrible environmental exposures. There's a town to the east here called Smithfield that when you drive through it, you smell the exposure from the CAFO that is there. Your tires will smell like that CAFO even when you get home to the garage in Raleigh. You have to spray them off if you drove through Smithfield. So these really are pretty dramatic sort of exposures. So we're interested in associations with immune-mediated diseases and that distance to these caged animal feeding operations. In particular, looking at gene-environment interactions with known genes that are associated to these disorders. So we looked at individual diseases as well as grouped diseases. And what I'm showing you here is a pretty dramatic example of a gene-environment interaction. So on the y-axis here, you've got the distance to the CAFOs. So this is how far away our participants live from one of those CAFO facilities. Our y has the odds ratio, so you're seeing our effect size. And here I'm showing you the sort of what the environmental effect is alone. And then I'm showing you that environmental effect and odds ratio by genotype for individuals with this, the homozygous minor allele. You see a very strong interaction effect. So we're interested in report these on multiple sort of genes in PTPN22, AHR pathway gene interactions with CAFOs exposures that were really convincing. So look at G by E. We're also interested in those geospatial linkages that I mentioned to you, and Kyle Messier that I mentioned there is really an expert in developing the actual estimates of exposure. So right here we're looking at air pollution. You can imagine this data comes from different EPA monitoring sites, and there's not an even distribution of those monitors. So we have to fit and estimate these different components of air pollution, and you can see a bunch of North Carolina maps with the values for those different components of air pollution. And he takes really sophisticated Bayesian modeling sort of approaches to associate those air pollution measures with sort of, in this case, the trainee was really interested in autoimmune skin diseases. And long story short, found some very strong associations when you looked at the mixtures. So if you looked at these individual components, you didn't see the effect, but when you're able to jointly model the different components of small particulate matter, air pollution was able to see this. And to give you a little bit of an anchor, I said I know I'm going through it quickly, but sort of the strength of these associations were about the same magnitude of whether or not you had a history as a smoker, which comes up in autoimmune diseases and many other common complex traits all the time. So this really is a strong magnitude of effect, like I said, on par with whether or not you smoke. Additionally, we mentioned we've got whole genome sequencing. And so we've been able to call the HLA MHC region to 6-digit resolution. And again, this is work from Eunice Lee, the woman that was doing the XFOS studies, and did MHC region mapping with adult onset asthma. So she tested population-specific SNP and HLA allele and then protein associations, and did it stratified for the 2 different genetic ancestry groups that we've got here. And long story short, she found some really interesting sort of population-specific effects. So really found that in sort of our participants with African-American ancestry, it was really HLA-B and HLA-DOB. I'm sorry, reverse that. In our European subjects, it was HLA-B and HLA-DOB. And that class 1 HLA genes were strongly associated with our participants with European ancestry, and it was actually class 2 genes for participants with African ancestry, which is interesting. And we're building on a lot of sort of the pipelines and work she developed for us for that HLA MHC analyses for stuff that we're interested in the All of Us cohort as well. Another really talented graduate student wanted to pursue GWAS analysis with adverse pregnancy outcomes. Like I said, I'm a human geneticist. I was incredibly skeptical about sort of the power of this study to do any GWAS mapping, and especially in sort of rare outcomes like adverse pregnancy outcomes. But it's one of those moments I'm glad I didn't listen to me and I listened to her, which happens to me all the time. But she was really interested in that. So did GWAS mapping in pegs of gestational hypertension. You can see we've got quite a small sample size when you consider sort of what you're used to for GWAS mapping. But here you can see that Manhattan plot down to the left. We've got a genome-wide significant hit in a couple of genes, and including this rare B gene. It's a retinoic acid signaling gene. We were able to replicate that in the UK biobank. You can see there sort of the lead SNP there. And really interesting, rare B makes a ton of biological sense. So it's been studied and shown to be associated with sort of severity of proteinuria and in preeclamptic studies, particularly had come up in the fetal genome, but we're seeing it here now in the maternal genome. So that paper was recently published, and she's going to continue other work in other cohorts with gestational hypertension and other adverse pregnancy outcomes. I mentioned the XWAS that we did on cardiovascular disease and warned you we were going to scale that up. So we did. So we took the same sort of approach where, again, we're looking across all of our surveys. Got that sort of summarized here in a graphical abstract. And we ran XWAS over all of our sort of common complex diseases that hit a certain prevalence that made us feel good about ourselves for statistical power. Cutoffs. So you can see it's the things that are high frequency: hypertension, cholesterol, migraines, asthma, type 2 diabetes, etc. And we pushed it through sort of what we now consider our XWAS pipeline. So we looked first at single exposure modeling, right? Analogous to a GWAS, I'm going to fit a regression model for each one of my exposures to each disease outcome. Then we followed up with sort of multi-exposure modeling. So really just sort of we use an algorithm called DSA, deletion substitution addition. It's sort of nice training, testing, validation framework for sort of lasso regression. And then think about sort of our results visualization and interpretation there. Here I've just got a cartoon of forest plots with different odds ratios. The other thing we wanted to look at in this study was also looking at the correlations across our exposure information, right? Because we know no one is exposed to one thing at a time, right? Back to that exposomics approach, right? Exposomics is everything's happening all the time, all at once, always. So could we look at sort of how these variables are correlated? And we didn't do anything all that clever, but we did something very, very tedious. And a theme to my lab is we do something tedious and probably take that too far. But we wanted to look at those correlations in survey data. We didn't invent any new statistical methods, but we did the really rigorous sort of hand cleaning that you need to do for survey data. So some survey data is binary, yes, no, you have this disease, you're exposed. Some of it is ordinal, right, where you've got, the equivalent of extra small to large t-shirt sizes in the data in the way that you've collected it. And if you want to be rigorous about correlations, those require different things. So a survey question that you have said, are you exposed to asbestos, yes or no? That's actually a binarization of an underlying quantitative value. And so if you just take correlation types for binary values, you're not going to get the right answers. So we did the tedious thing you need to do of fitting polychoric correlations and, like each one of those data type by data type across those thousands of exposures. So ran that correlation analysis. We also know different surveys have different sample sizes, and a correlations interpretation is heavily influenced by sample size. So we fit a BLUP model to actually shrink correlations so that we could put them on the same scale there and help visualize those results. Here you're seeing sort of a spaghetti plot here, right, that will represent the correlation across different variables. And what we really wanted to do was make these results as available as possible. So we ran these XWAS for all those different outcomes, and we keep building on the diseases that we've evaluated. And we put all that data available in what we call a PEGS Explorer. So if you ever wanted to Google NIEHS and PEGS, it takes you to our website where you can get information on the study itself, but also go into some of our results Explorer. So we've got an interactive web tool, if you're interested in this, called PEGS Explorer, and you can pull up for any of the diseases and any of the questions that we have asked. Here you're seeing an example. You can interactively go through each one of those results, see what the association effect size was. We've got options for you to pick that in different strata of the data, with or without different covariate adjustments. And you can also download, we've got a visual toolkit that you can look through, and you also can download all that data and deal with it computationally if you're interested. So we keep building on that. Like I said, then we've got these correlation globes as we show them that let you interact with how people are exposed to these different exposures jointly. That's being a little weird with there we go. And then the last study I'm going to talk about here in PEGS actually gets more towards the precision health that I know this group is interested in. So thank you for your patience in some of the discovery stuff that we've done. We'll talk about a recent study with a clear precision environmental health aspect, and then what we're doing in that space right now that's ongoing work. So I've had an interest in diabetes, have done a lot of pharmacogenomics with diabetes drugs, and honestly, the strongest signals we see in PEGs on any of those common complex traits tends to be diabetes. So here we wanted to directly compare sort of the performance of polygenic scores, which have been the focus of human genetic, like a focus of a lot of human genetics work in the last decade. At least, right? Where what you're trying to do is across the genome, whether that is a certain number of genes or really you're trying to fit all of the genetic variants across the genome and build a predictive score for your risk of a certain disease. So we're interested in comparing polygenic scores that have gotten a whole lot of work with polyexposure scores. So hopefully you can imagine what we mean by polyexposure score. So analogous to a polygenic risk score, we're going to look across those exposures and build a risk score that sort of is associated and predicts your disease outcome based on your exposures. So our first example here is in type 2 diabetes. So I'll try to go through slightly complicated plots pretty quickly. But so we compared a polygenic score. There's a catalog of those available. I'll talk about that a little bit more in a minute. So there are some sort of well-studied, well-used polygenic scores. We fit our own as well to compare. And then we built polyexposure scores from our survey questions. We also built a clinical risk score for diabetes that has been in the literature. And this clinical risk score includes things like, have you ever been diagnosed with prediabetes and BMI? This is a very, very strong predictor. We did our XWAS to help explore some of those exposures, and then built a poly exposure score, again, with really pretty simple sort of lasso-based methods with training testing. We found some interesting things in this study with asbestos and coal dust. And then sort of long story short, what we showed is if here's my odds ratio of each of those scores. So I've got my clinical research score, my polygenic score, then combinations of those, my environmental risk score. Long story short, the environmental risk score outperforms the polygenic score summarily. And even when I fit every one of those different combinations, the poly exposure score always adds significant value, whether I want to look at that at AUC or an NRI or whatever that fit is. So even if I'm modeling with a clinical score and a polygenic score, adding that exposure score adds value. Our ongoing work, and I'll show you a little bit of hot off the presses data that came off literally this week to present, but we're following up our XBOS approach with the growing number of GIS exposures. We're interested in some genetic correlation analysis as we think about sort of the association between variants and exposures. We're using that epigenetic data for epigenome-wide association studies. We're also following up with epigenetic biomarkers. So there's a lot recently out there on sort of aging clocks, right? Your biological age versus your chronological age based on epigenetic data that we're working on. I'm doing more integrative omics analysis. And then what I'm going to talk about here for a minute is the expansion of that poly exposure and polygenic comparisons. So as I mentioned earlier, stole a graphic here for polygenic risk scores, right? For across your genome, you can put sort of the population you're interested in percentiles of risk, right? The 5th percentile versus 99th percentile, the same sort of interpretation you would have for test scores or anything else. So thinking in that way, we build the poly exposure scores similarly. So like I said, this poly exposure score, it's a composite metric that aggregates these multiple types of exposures that you might see, designed to capture the broad and cumulative impact of diverse, potentially modifiable factors on health or other outcomes, and wants to emphasize the interplay of various exposures rather than just one single, right? So really taking that exposomics, that joint exposure, and what does that do for your risk? And in this study, differently than the diabetes one that we published, we've broken our poly exposure scores into 2 types. So hang with me on jargon, because some of the jargon I'm trying to invent. You can imagine some of our exposures are things that are well outside of any individual participant's control, place-based disparities, things like social determinants of health. And we've built scores, and we're calling them polysocial scores, because these really are aggregating social determinants of health that an individual can't normally change. You can't tell somebody to move to a better neighborhood. They already would have if they could have, right? So these are place-based disparities, things like income and socioeconomic status, housing types and conditions, social vulnerability, sorts of metrics. And we contrast that to what we're calling in this study our poly exposure scores. So these are things that might include things that really might be interventionable at an individual level. Those occupational, residential, hobby-based exposures that you could think about buying new filters for your house or mediating some of that exposure. So think of them as more of these interventional factors at an individual level. Occupational hazards, hobbies and lifestyle choices, and stress exposures that are also modifiable. So we started by calculating polygenic scores. So there's a catalog, there's a polygenic score catalog that has a number of polygenic scores that have been previously published in other studies. There are a little over 3,000 of those that were published. So we downloaded those and fit every one of those to our PEGS participants. So we've got these multiple polygenic scores for each one of our participants. We looked at those common complex traits that are again above our prevalence cutoff. So at the things that you saw for the exposome-wide association studies are now being used in this. And you can imagine that these different traits have a different number of polygenic scores that have been published. Things like type 2 diabetes that are more studied had about 80 that we downloaded. And things like migraines only had a couple of published polygenic scores. So keep in mind, there are a number of caveats to that, but these were picked based on the closest relevance to our phenotype. And then we actually, when I'm going to show you results, we're presenting the results of whichever polygenic score actually had the highest AUC in PEGs. So we're trying to give these polygenic scores a heads up, like a head start, right? So of those almost 80 that we fit for type 2 diabetes, I'm going to present the results of the best fit in PEGs, and then compare that to our poly exposure scores. I'll go by this quickly. If you're familiar with an upset plot, it's really easy to interpret, and if you're not, it's a little hard to explain, but it's sort of a fancy Venn diagram. So our variables that came together for the polysocial score versus the polyexposure score, we had a lot of overlap, and the same sort of social determinant measures came up across diseases. Then for the individual exposures there to the right, you see fewer connections down below across diseases, that more unique exposures were pulled in. Overall, again, we did the same sort of let's compare our polygenic scores to these two poly exposure scores. I'm going to draw your attention to this graphic here. Again, on the X, you've got the traits that we've studied, and on the Y, you've got the AUC. So the higher the dot you see on this dot plot, the stronger the association, the stronger potential prediction. And I've got sort of the different scores colored here. So the purple are our polygenic scores, the orange are the polysocial scores, and the green are the polyexposure scores. Long story short, what I want you to see here is in every case, the polygenic score has much lower performance than either the polyexposure or the polysocial score, and there's really not a big difference between the polysocial and the polyexposure score. This becomes important when I talk to Jan, that's my co-PI in PEGS. She wants to know not only will these exposures predict the outcome, but what's the minimum number of questions I can ask, right? Our PEGS participants answering over 1,000 questions isn't ever going to be practical for any amount of translation, but can we get down to sort of a minimal number of sets of questions, which is interesting. I'll show you here just a couple of other results if you're used to seeing sort of an AUC or model fit in a different way. Here's our example for type 2 diabetes. Again, the purple is the polygenic score, and all these others are our different polyexposure scores. And you can see in lower GI polyps, you see even a stronger gap there between the polygenic scores and the polysocial. I think it's promising. Showing it in one other way just to try to drive the results home. Here you can look at sort of prevalence plots. So this actually is a number of cases. It's prevalence of that disease given the different percentile scores for the different scores. So across a polygenic score, polysocial, and polyexposure. And what I want you to see is that there's more discrimination. You get a higher range from bottom to top there with the polysocial and polyexposure scores than you do the polygenic scores. So I'll stop there. I know that was a whirlwind tour of things I've been thinking about with PEGS, so thank you for your patience there. I'm going to switch gears now to talk about the partnered research study that Josh and all the leadership at All of Us has been just incredibly supportive and amazing on. So talk about that a little bit. So stealing some slides from them, I don't even know who I stole it from, but it's probably from you, Josh, on sort of priorities in All of Us of the different types of data that are integrated, right? There's a wealth of information in genomics and wearables, the biospecimens y'all have collected combined with a health record, different behavior, and especially fantastic social determinants of health surveys, and then a number of physical measurements, how to sort of pull in more and more environmental data. I find personally really exciting. And doing it for all the reasons, right? Sort of pulling that sort of data in helps how do socioeconomic factors interact with exposures for risk, to really think about health disparities. Think about risk and prevention. What environmental biomarkers could identify risk for future disease? Could these sort of biomarkers be helpful in diagnostic? What sort of exposure signatures do you find in patients with this or without this different condition? Treatments and outcomes. Can we objectively assess whether treatment or intervention might be effective, right? Sort of, there's so many broad goals that you could think about when you start thinking about pulling environmental data into All of Us. We had a fantastic workshop. almost 2.5, almost 3 years ago right now, where all of us leadership, folks from NIEHS and people from the community at large, really spent a few days thinking about sort of priorities when you think about adding in environmental data. And a number of themes emerged, sort of linking to geospatial estimates, linking to climate and weather data, exposomics, whether you mean that from surveys or mass spec-based sort of exposomics technology, thinking about personal exposure risk and effective exposure on diverse populations. And we followed that up with some really exciting studies to incorporate the environment into all of us. So really, we sort of left that with sort of a three-phase approach to incorporating environment. For phase 1 and 2, we're really based on geospatial estimates. And all of us has made just an absolutely incredible investment in getting addresses, address histories for participants, and then doing the geocoding to link that data to exposures. They're doing that through a CLAD mechanism, and we're hopefully having some collaborations with another NIEHS effort that I'll just mention briefly. And then phase 3 is the partnered research study that I'm leading here. Where here we did sort of a pilot case cohort study looking at incident diabetes. So actually able to collect sort of untargeted mass spec-based exposomics in biospecimens from participants that had incident diabetes versus a cohort sample for comparison, which is really exciting. We're in the middle of that data collection right now. I had a meeting yesterday. We're done 51 out of 71 batches of collecting the exposomics data. So it's moving along nicely. Oops. I mentioned for the geospatial estimates, there's another effort at NIEHS. CORDS is the acronym. The acronym is under development again right now, but it's a the CORT-F funded project to build tools to do geospatial linkages. So it's taking data from NASA, from EPA, from satellites, imagery, and building tools to help people do those linkages, both to do software, to build and curate web-based resources. So let's say you're a human geneticist that's never done environmental data before. Been there, and you say, maybe I'm interested in air pollution. It's building web tools and AI tools to say, I'm interested in air pollution, and it's going to pull up what resources are actually accessible to you. Here's the EPA and how they do it. Here's NOAA and NASA and their versions, right, to get you started on adding environmental data into your studies. And I said specifically, this All of Us ancillary study launched last July. We're working on we identified participants, sent biospecimens to a laboratory here in North Carolina that runs these mass spec sort of assays, and that data is in progress now. So we'll have that on the case cohort study. It will go into the All of Us data research center and their ecosystem. So that data will be available to everybody that has access at the right tier within All of Us, which is really exciting. I said it here. Sort of moving on phase 3, where I said we're looking at sort of that exposomics and associations with outcomes. You can imagine we'll be doing things like testing for associations of those exposomics biomarkers with development of diabetes, with the development of some of the diabetic complications, retinopathy, any of those others, with comorbidities and comortalities. And we'll have the wonderful opportunity to sort of look across omes. This is one of the largest exposomic studies ever done. So just having this number with this geographic and demographic diversity is pretty unprecedented and pretty exciting. So we're in the middle of that right now. The mass spec technology that we've chosen, it's the HERE lab. It's here up the street in Chapel Hill. Collects a broad panel of metabolites, whether you want to call it metabolomics or exposomics. So we'll have a number of sort of endogenous metabolites representing host metabolism, so amino acids and polyamines and carnitine, these sort of host metabolites. Then we'll have a lot that we can measure in the food metabolome, caffeine, and all sorts of different sort of subcategories of vitamins, minerals, etc. A number of environmentally relevant metabolites, PFAS, phenols, parabens, phthalates, things you probably heard in the news, if nowhere else. And then additional categories, we'll be able to look at drugs and medicines, folate, vitamins, amino acids, etc. This is what we're expecting off there. So we can be thinking about things that both use the annotated metabolites that we'll be able to measure, as well as thousands of sort of quantified metabolites that don't have any annotation to them yet. And this can support a wide variety of things. So here's a very big slide with right now in all of us, we're going to have genomic data, exposomic data, we'll have society data, societal data, social determinants of health, and then phenotypes. So you can imagine, hopefully, some of the things I presented that we've done in PEGS, I hope some of the similar thinking can come into all of us wherever we're thinking about XWAS, whether that's from the biochemical data or the geospatial data, can think about risk score analyses, again, using all of these different components. And to mention the long read sequencing has epigenetic markers for all of us too, so you can add that as well. You can think about all the sorts of interaction analyses. And then I think a lot about variance decompositions. How much of this disease is due to genetics and environment? And it's really exciting. Like I said, I've had a whole lot of fun in PEGS, and it is cute and adorable. And being able to think about some of these approaches and something that has the mega sample size of all of us is just really, really exciting, because that's what it's going to take to look at gene environment interactions. So all of this and more will be possible, and I think that's really exciting. And also, I'm in it's biostats and biology. But I'm suddenly echoing. I don't know why, but like I said, a lot of this is going to need to be fueled by methods development, right? There aren't tools out there to ask and answer all these questions. So there are a ton of really exciting data science opportunities as well that I think are important. So I'll stop there. Again, thank you for dealing with this whirlwind. I hope it was informative and sort of the ways we're thinking about this. Of course, I have to thank all sorts of people, and there are more people to thank than just listed here, but of course have folks with the PEGS leadership that I mentioned. I see somebody already out there from our external advisory board, even that I see in the audience. I said people with the GIS, all of our colleagues at all of us that are just amazing. I said highlighting some of the computational work, some of the bioinformaticists that have done a lot of this, and our collaborators and contract support. And then first and foremost, the PEGs and all of us participants that give us generous access to the data. So I'll pause there for questions, and I'll stop sharing. There, thank you.
I don't know if you could hear that, but people were clapping for you. You could probably at least see them clapping, perhaps. So now let's open to questions. I have, as people, you can write them, if you're online, just write them into the Q&A. And if you are here, you can just come up to the mic. The first, I'll ask first one that I got online, and it was, how can we bring this knowledge to the clinics?
Well, I mean, like I said, we're in sort of the discovery phase, right? The poly-exposure scores and things, but I hope that will actually directly be clinically applicable. I hope at some point, and there are some huge and interesting efforts on adding social determinants and other exposures to electronic health records, actually, but I'm hoping this sort of information, this sort of knowledge could help clinicians learn better who to screen for what diseases, or which questions to ask and how to take that into more holistic care. So I know we're a long way from that, but I hope we're less far away from that than further, if that helps.
Yeah, it makes me think of the all the sections of our notes and talking about social history, how we could really expand that as well. Another one online is, do you have sufficient data for the vast numbers of variables to use deep learning methods to enable more agnostic results?
Yes. Do we have the sample size, right? Anytime you ask a statistician if they should have more samples, we will say yes. It's sort of like asking a dog if they're hungry. Like, yeah, the dog's hungry and I would love more samples. But we absolutely are able to use some of those new machine learners. And I've got some folks working on that. I always think of, right, anytime you're doing hypothesis generation, I want to know what would be a useful hypothesis to follow up. So it meant like, my dissertation 20 years ago involved neural nets. I've been doing that sort of machine learning for a really long time. So you have to think there are cases where that sort of prediction, that sort of multi-omic integration is really useful, and could be sort of predictive power. We've got some folks working in that space with that sort of machine learner. And then I sometimes contrast that to the stuff that maybe I was more heavy on here, where we're trying to do hypothesis generation that we could follow up. Right, back to the clinical question, we're long way away from somebody spending tens of thousands of dollars for multi-omics in a clinical setting. So we have one avenue that I haven't talked about a ton with machine learning and with methods to narrow down the search space for G by E follow-up that's more targeted, that relies heavily on machine learning. But yeah, we can wield those tools in this sample size for sure.
Great.
Hi. Thank you so much for your talk. I have a maybe not completely formed question, but
Love it.
in looking at all of the awesome survey data that you guys have, I think I first thought about how you get people to answer those surveys, maybe particularly people who live in areas that don't have great internet access or things like that, because I went to college in North Carolina, and something we talked about a lot was the poor health outcomes in the western part of the state. So just wanted to see if you could touch on that and maybe talk about any challenges you've had with like follow up with participants from those areas and things like that.
Absolutely, yeah. It's absolutely a challenge. So a lot of the recruitment was done with, at the beginning, when PEGS was starting, was done with very targeted recruitment drives. And so that involved things like partnerships with churches in those like rural areas that are a community that is trusted and that could interact. There were also a lot of joint recruitments in other initiatives, a lot of like women's health sort of initiatives. So some of it we have more women because women tend to volunteer for things, but we also had some recruitments tied into some women's health initiatives. We had massive challenges during COVID, like a lot of people, of people moving and relocating. We do a lot of follow-up electronically now, right? Like we can take your addresses and like every like there are plenty of, computational resources that track who's where and can reach back. So we definitely had a gap in COVID with people moving and sort of reaching back out as well. For the most part, I'll make, I'm gonna sound really flippant, but I'm not. These participants will just volunteer for anything. Like our participants are just amazing. I don't know, like I almost worry about them sometimes. Of like, sure, okay, you'll keep going. So also now we've switched a lot to electronic, like we're entirely electronic surveys in sort of our ongoing follow-up sort of callbacks, where we incentivize people to take the surveys, whether it's with Amazon gift cards or other sorts of sort of simple things. The other thing I will highlight about PEGS that I haven't mentioned and just use this as an excuse is all of these participants are available to call back in our clinical center at NIEHS. So PEGS has also served as almost a core facility for people that need biospecimens or specific things. We've had people do studies where we've called PEGS participants back in to do skin biopsies on sun-exposed versus unexposed regions and looked at mutation rates. We've had participants call back for inhalation assays. Like they'll sit in a body pod and breathe diesel because we asked them nicely. So it's a really engaged community that cares a lot about it. So in some ways I'm like, I don't know the secret outside of like these people are just really, really generous with their time and energy.
Awesome. Thank you.
Another one online. Hi, Alison, great talk. Does PEGS have disease incidence data or only prevalence? Are there efforts to bring in longitudinal data to PEGS?
Yes, right now we have prevalence. It has been an ongoing effort, but it's maturing. We are linking to health records, first with UNC Chapel Hill. So once we're there, we've done a lot of the data sharing. We've had a lot of IRB and reconsent to get this done, but I'm hoping in 2025 we'll have electronic health records. And UNC Chapel Hill doesn't just have, their health system isn't just the hospital, but like a large number of primary care places are included in that UNC. So it'll suddenly have longitudinal data sort of in the same way that all of us does as we work to finalize that health record. So I'm very excited for that.
Les.
Great talk, Alison. Les Biesecker. Question for you on how this really goes forward. My, head explodes with the number of variables you're talking about here. And, and of course, in genetics, it's taken us 15 to 20 years to go from GWAS associations to people actually starting to work out cause and effect of these variants. It's been a really hard slog. And I can't even begin to imagine what that equivalent looks like for you from this polyexposome score to get to cause and effect, or what it is in that score that is disrupting the physiology that makes these people sick. What are you thinking about what that looks like?
Yeah, I understand and have no no easy solutions there, right? And I know you don't think we do. Yeah, like I think some of the issues there's some things that are way easier in genetics, right? Like I know how to format that. Like I've got nice structured data. It took us a while in the GWAS era for chips to evolve and get to like there were challenges, but I can harmonize genetic data from the UK, from Canada, from all of us trivially by comparison of harmonizing environmental data as well. So there are all sorts of issues with sort of data sharing in exposomics and in environmental health sciences. Like I said, that PEGS Explorer, some of those things, like for the most part, if you want an epidemiological study, there's so many data use committees and people have already like pinned what their little niche is for this area. So I think data sharing is going to have to be more expected for us to get there. A more open-fisted approach that I think genetics has had more than environmental health sciences. And I don't trivialize that there are different issues to consider with that. I'm not trivializing that challenge. But I think that needs to happen. I think the opportunities for people in leadership at all of us in UK Biobank and other international efforts, like a willingness to try to standardize at that level would go very, very far for that like G by E or E is notoriously hard to find, right? For all the statistical challenges and all the high dimensional challenges. And honestly, the mega sample size is what it's going to have to take to get there of just these huge mega samples with then clever people doing causal inference and observational studies to help narrow that down, and always really clever partnerships between basic biologists, mechanistic biologists, toxicologists, whatever. Yeah, like it's just going to be even more interdisciplinary to get somewhere meaningful. Is that helpful or sort of in line with what you're
it's very analogous to what we were saying when GWAS started. Yep. It's like,
yep.
Oh my God.
Well, I said it.
I'm going to take a prerogative to ask a follow-up question based on that I'd written down, which was, it's interesting how similar the polysocial and the polyexposure scores were in predicted value, one being intervenable, one not. I was wondering if that's actually like just similar predictive values, or it's actually similar for the same person. And if so, what does that actually mean? Does that mean that we tend to cluster our exposures in some way, that are the ones we intervene on not, or, is there something we can learn from that basically?
Yeah, we're just exploring this. Like some of this is hot off the presses. I would say it's more surprisingly the same predictive power than necessarily the same individuals. They are certainly correlated. Absolutely, there is correlation, but the predictive powers are more similar than I would have expected when I just looked at the correlations. But we're really just starting to pick all of that. We're starting to disentangle this, not just for diabetes, but more broadly. But yeah, the predictive performance is stronger than I would expect on the correlation.
Neil, I think you get the last question.
Oh, this one will be relatively simple. So if I heard you right, you said that you were in 99 of the 100 counties.
Mm-hmm.
What is it about the one county that you're not in?
I'm not going to remember the name of it. I'm from North Carolina, so I should. It's actually the one to the furthest northern right that is just really rural. It's like right at the edge of the Outer Banks. There are not many people there. And we
Yeah.
haven't.
Neil wants to go recruit in that county for you. So,
But if I need to go to the
connect
beach,
you
if I
afterwards.
need to go to the beach to really make sure we're there, I would make that sacrifice. I would go to the beautiful beaches if I had to.
Great. Thanks for all the folks that asked questions online, and it's also, it's been a great talk, Allison. I really appreciate you coming and giving this. I think you give a exposure to a kind of thing we don't get to talk a lot about, and the importance. We often talk about the importance of zip code and genetic code, but this is like a whole different layer on that as we talk about lifestyle, environment, and biology here. So thank you very much for joining us.
Thank you. This is lovely.
And we're clapping one more time if you can't tell. Thank you.
Thank you all.